Whether it's a barely detectable speck of DNA from a decades-old cold case, like those that haunted investigators seeking the Grim Sleeper, or a fragmented piece of genetic code extracted from the 5,300-year-old Ötzi the Iceman, analyzing challenging DNA samples requires a shared arsenal of powerful lab techniques. Welcome back, molecular adventurers. After exploring how these crucial samples are meticulously collected in our previous 2 episodes, we now step inside the lab to explore the common ground between forensic science and paleogenetics. Both fields frequently face the daunting task of analyzing DNA that is present in minute quantities, heavily degraded, or even contaminated. Let's look at the fundamental techniques that help unlock these genetic puzzles. Before we dive into the solutions, let's understand the problems. Both forensic scientists working on trace evidence and paleogeneticists studying millennia-old remains face remarkably similar hurdles when it to DNA analysis. First, there's low copy number LCN DNA. This means there are very small amounts of DNA present, sometimes just a few cells. Think of touch DNA from a doorknob in a crime scene, or the minuscule amount of surviving DNA in an ancient bone fragment. Such tiny samples are inherently difficult to work with. Then we often contend with DNA degradation and fragmentation. Over time, and due to environmental factors like heat, humidity, or UV radiation, DNA molecules break down into tiny pieces. Imagine trying to piece together a shredded document. That's similar to working with fragmented DNA from an old cold case or an ancient skeleton. Adding to the challenge are PCR inhibitors, substances found in various samples like soil or certain fabrics that can actively block our genetic analysis. And of course, the ever-present threat of contamination looms, as even a single foreign DNA molecule can ruin a sensitive test. The journey to analyzing challenging DNA begins with extraction, carefully isolating the genetic material from surrounding cellular debris and potential inhibitors. For these tough samples, standard methods often aren't enough. Specialized protocols are essential. In forensics, extraction methods are adapted for various evidence types. For example, in sexual assault cases, a differential extraction can separate male sperm cells from female epithelial cells. For other trace evidence, specific kits are designed to capture every last bit of DNA. For ancient DNA, the challenge is even greater because the DNA is already highly fragmented. Paleogeneticists use optimized methods, often silica-based extraction techniques, which are incredibly efficient at binding even short, broken DNA fragments. The goal is to maximize DNA recovery while minimizing any further physical shearing of the already delicate ancient molecules. After extraction, quantification and quality assessment are critical, often performed using quantitative PCR or qPCR. This tells us not only how much DNA we've recovered, but also gives us an initial idea of its quality and how degraded it might be. This information is vital for deciding the best strategy for the next step. Once we have extracted the precious DNA, the next crucial step is amplification, making many copies of the target regions so we have enough material to analyze. For challenging samples, standard PCR that we've covered in other episodes often isn't enough. We need to bring out the power-ups. One such power-up is Multiplex PCR. This incredible technique allows scientists to simultaneously amplify multiple DNA targets in a single reaction. For forensic scientists, this means analyzing many different STR loci, those unique repeating DNA sequences used for identification, all at once. For paleogeneticists, it allows them to target various genetic markers, maximizing data from tiny samples. Crucial for working with degraded DNA is MiniPCR, or using short amplicon design. This means designing our PCR primers to amplify very short stretches of DNA. When DNA is broken into tiny pieces, amplifying these smaller fragments dramatically increases the chances of successful amplification. It's like trying to read small surviving sections of that shredded document we mentioned earlier. This approach is absolutely vital for ancient DNA and older forensic samples. Another powerful tool for highly degraded or extremely low copy number samples is focusing on mitochondrial DNA, mtDNA. Unlike nuclear DNA, which we have 2 copies of per cell, Mitochondrial DNA can have hundreds or even thousands of copies per cell. This much higher copy number makes it a goldmine for analysis when nuclear DNA is too degraded, such as in hair shafts without roots, very old bones from both crime scenes and archaeological sites, or even very old plant material. mtDNA is also especially useful for tracing maternal lineages. For the most extreme cases of low quantity or severely degraded DNA, forensic and paleogenetic labs turn to even more specialized PCR strategies that push the limits of detection: nested PCR and booster PCR. These aren't just power-ups, they're a targeted strike. Nested PCR. This is a highly sensitive, two-round amplification method that provides exceptional specificity. First round, outer primers. We begin with an initial PCR reaction using a set of outer primers. These primers amplify a somewhat broader DNA region that encompasses the specific, very tiny segment we are ultimately interested in. This initial round produces a preliminary pool of amplified DNA. Second round, inner primers, nested. Then, a small amount of the product from that first PCR reaction is used as the template for a second, separate PCR reaction. Crucially, this second reaction uses a new set of inner primers. These inner primers are designed to bind within the DNA region amplified by the first set of outer primers. By essentially nesting the second reaction inside the first, we dramatically boost the amplification of our specific target while simultaneously eliminating non-specific products. Contamination note. Traditionally, transferring the product between rounds can risk contamination. Modern techniques like single-tube nested PCR minimize this by using special primers that are only released after the first round, all within the same tube. Booster PCR. This technique tackles the problem of very low template DNA by cleverly adjusting primer concentrations between stages, which reduces nonspecific amplification and primer dimers. Stage 1, low primer concentration. The first stage uses a very low diluted concentration of primers. This approach is critical because it prioritizes specific amplification of the rare target DNA molecules while minimizing unwanted reactions between primers or nonspecific binding, which can overwhelm a tiny signal. Stage 2, standard primer concentration. After a few cycles in stage 1, the primer concentration is increased to standard higher levels. By this point, the specific target DNA has already been preferentially amplified enough to outcompete nonspecific products. The higher primer concentration then allows for efficient and robust amplification of the already enriched target DNA. Both nested PCR and booster PCR are indispensable tools for pushing the boundaries of what's possible with the most challenging degraded or low copy number DNA samples, whether from ancient mummies or trace crime scene evidence. From carefully coaxing DNA out of ancient bones and mummies to amplifying the faintest traces from a crime scene, these shared molecular techniques form the backbone of how we unlock genetic information from the most challenging samples. They allow us to peer into both the distant past and the crucial details of a crime. Once amplified, these genetic products are ready for the final stages of analysis and interpretation. Next time, in Part 4, we'll step onto the forensic lab bench specifically, delving into the specialized techniques and analyses used to interpret DNA profiles for individual identification and to bring perpetrators to justice. You'll see how the amplified DNA is transformed into the undeniable evidence presented in court. Make sure to subscribe to the Geneticist, like this video, and ring that bell to catch all our deep dives into genetics. See you soon!